Three basic parts of every cell. Is a cell really just a water balloon?
Okay, so basically, a cell is just this. One balloon, some water, done. End of biology.
Convenient. Then split it in half right now. A real cell does.
Fine, give me 30 seconds. Loser buys samosas after class.
Before the samosa bet, one question. Can your balloon choose what goes in and what stays out?
Okay, so basically, yes, things enter when I pour them in. Obviously, the balloon picks, sort of.
So, a sealed balloon at recess. Alive or dead? Pick one.
A real cell has a gatekeeper, the membrane. It picks essentials in, escorts waste out every second.
Right, gatekeeper, knew that. But inside, it's just water, yes.
Inside the cell, cytoplasm is the workshop. Proteins assembled, sugars burned, repairs running, all at once.
Workshop. Your balloon water has done what exactly? Sat there?
And one more thing. Every cell has a control room, the nucleus. It tells the workshop what to build, when to divide.
So my balloon isn't a cell. Skin, sure, but no gatekeeper, no workshop, no control room.
Cell membrane, the gatekeeper. It lets in glucose, blocks toxins, ushers waste out every second of every day. Cytoplasm, the workshop. Tiny machines work shoulder to shoulder inside, never clocking off, never empty. Nucleus, the control room. The blueprint lives here. Every order to build or divide issues from inside.
Gatekeeper, workshop, control room. 3 parts, 3 jobs. The cell is a tiny city, not a balloon. Yeah, okay, I owe Zara samosas. The balloon was just skin and water, nothing more. If we ripped the membrane, what dies first?
A cell is not a water balloon. 3 working parts run it. The membrane is the gatekeeper, letting essentials in and waste out. Cytoplasm is the workshop. The nucleus is the control room. Plant cell versus animal cell. The extra structures. Should both clay cells get the same wall?
Free period. 2 clay cells, 1 tree. Mine, tomato. Yours, cheek. Race.
Okay, so basically, both get walls. Every cell has a wall. Brick wall biology friend.
Convenient. Then you'd never blink. Stiff face, glued grin, dead by lunch.
Test the claim before you mold more clay. Squeeze a ripe tomato. Now, pinch your own cheek.
Tomato form, cheek very soft. Wait, so cheek cells have no wall at all?
Right. Animal cells stop at the membrane. Plants add a thick wall on top of it. Cellulose, rigid, strong.
Then bricks come off the cheek model. Now, lab rule. What you can't squeeze, you don't build.
Fine, but the green dots in tomato leaves, they show up in my cheek too, no? Some glow?
Those green dots are chloroplasts. Plant only. They catch sunlight and cook sugar inside the leaf all day.
And the big bubble in the center of every plant cell, that vacuole. My tomato gets it. Your cheek does not. Animal cells don't have a cell wall. Plants do. Plus chloroplasts, plus one large central vacuole.
Cell wall. Cellulose, rigid, brick-like outside the membrane. It locks the plant cell into a fixed boxy shape. Chloroplasts, green solar panels packed inside. Sunlight enters, sugar leaves, all through daylight hours. Large vacuole, one fluid-filled bag, nearly half the cell. Stores reserves, holds water, pumps the plant turgid.
So plants get cell wall, chloroplasts, large vacuole. Animals get the membrane only. 3 extras.
3 extras? Tomato got the upgrade pack. My cheek went basic plan.
Tear the bricks off your cheek model. We're not turning you into a cardboard puppet. Where would chloroplasts hide in your skin? Anywhere?
Plant cells add a rigid cell wall outside the membrane. Chloroplasts let plants catch sunlight and cook sugar. Animals never carry them. A large central vacuole stores reserves and keeps the plant cell turgid. Cell shape matches cell function. Are textbook cells really all just square dots?
Look! Muscle, nerve, onion, all the same square dots. Microscope just zooms.
Square dots. Right. So your nerve runs from spine to toe in one square?
Many squares, lined up, same shape, just lots. Bet you are fruity.
Here are 3 slides. Muscle, nerve, onion. Look once. Then tell me they're the same square.
Okay, so basically, first one long and stripy. Second one branchy. Third, bricks.
Stripy, branchy bricks. 3 different shapes. Square dot theory, officially dead.
Each shape fits the job. A muscle cell is spindle-shaped, long and tapered, so it can contract and pull.
Contract like pulling a rope. So the shape helps the muscle do the squeeze?
Exactly. A nerve cell stretches long with branches at both ends, like a wire with antennas, carrying signals across the body.
Onion cells. Why bricks?
Onion cells are rectangular. They stack like bricks in a wall. Plants need that stack to stand upright. Shape, function, stuck together.
So cells don't look the same. Shape matches what each cell does. Spindle, branched, brick, not square dots.
Muscle cell. Spindle-shaped, long and tapered. Built to contract and pull a load with every pulse. Nerve cell. Long with branched dendrites. Built to carry signals from your spine all the way to your toe. Onion cell. Rectangular like a brick. Stacks tightly with neighbors to give the plant a stiff structural wall.
Spindle, branched, brick, 3 shapes, 3 jobs. Shape isn't decoration, it's the design itself.
Toss the square dot theory in the bin. Your fruity is officially overdue.
If nerves weren't branched, how slow would touch be?
Different cells look different. Shape is not random. It tracks the job. Spindle muscle pulls. Branched nerve signals. Brick onion stacks for structure. Each cell's shape is the design clue to what that cell does. Preparing and observing an onion peel slide. Does the slide really need a paper-thin peel?
Lab practical. 2 slide trays, 1 grade. Mine, onion. Yours, onion. Begin.
Okay, so basically big chunk, drop of any stain, cover slip on top, done in 20 seconds.
Convenient. Then look through it. Light gets through your chunk?
Before grading, Arav, hold up your peel. Now hold up Zara's. Tell me what the light does.
Mine opaque, white. Hers see-through, paper thin.
A microscope needs light passing through. Peel the thinnest single layer, the inner film, not a chunk.
Right. But the stain, any color, no? Anti-rust paint should work fine.
Anti-rust. Sure. Why not motor oil while you're at it?
Safranin, that red dye on the side bench, it binds cell walls and the nucleus. So each rectangle stands out under the lens.
Cover slip, that I know. Drop it straight on, squish out the bubbles like a paratha.
A slide is not a chunk, and any stain and a slap. Thin peel, safranin, cover slip at 45. That's the rule.
Step 1. Peel the thinnest film of onion, the inner skin between two layers. Light must pass through cleanly. Step 2. 1 drop of safranin stains it red. Cell walls outlined. The nucleus shows as a dark dot inside every rectangle. Step 3. Lower the cover slip at 45 degrees. The angle chases out air bubbles before they ruin the view.
Thin peel, safranin stain, 45-degree cover slip, microscope ready. Red bricks, visible at last.
Okay, my chunk goes in the bin. Slide attempt 2. The right way this time.
Your old chunk under my microscope. Black square, no cells, no win. If we skipped safranin, what disappears from view?
Peel the thinnest film of onion so microscope light passes through. 1 drop of safranin stains cell walls and the dark nucleus dot. Lower the cover slip at 45 degrees to chase out air bubbles. Ostrich egg yolk, the largest single cell. How many cells are inside a single yolk? One ostrich egg, staff room bench. Guess how many cells make up that yellow yolk? Closest wins lab pairings.
Okay, so basically that yolk is huge, so 10 million cells? Maybe a billion? I'll write a billion.
I'm writing 1. Just 1. If I'm wrong, you pick lab partners. If I'm right, I do.
1? Zara, it's the size of a mango. Every visible blob of biology is many small cells. That's the whole rule.
Says who? Show me the rule. A cell has a membrane, a cytoplasm, a nucleus. Nothing in there says small.
She's pulling on a real thread. The definition lists parts and jobs. Gatekeeper, jelly workshop, control room. Never a size limit?
Fine, but onion peel cells, bricks. Cheek cells, polygons. All tiny, microscope tiny. Where's your giant exception?
Prove it. Crack the egg. Inside that shell, there's 1 yolk, 1 membrane around it, 1 jelly inside, 1 nucleus somewhere.
There. Look. 1 yolk. Intact. A thin film holds it. Measure across 170 millimeters.
Wait, that whole orange blob is 1 cell? Not a million packed together? My billion is mildly off.
Mildly. Bacterium to ostrich yolk, size spans a billion times. The rule was never small. The rule is 3 parts.
Arav said visible blobs must be many small cells. Wrong. 1 yolk, 1 cell, held in your palm. Size was never the rule. The thin film around the yolk, that's the cell membrane, the gatekeeper. It chooses what enters and what leaves.
And the yellow jelly itself? Cytoplasm, the workshop. Every life process inside this cell runs in that goo.
And tucked inside, one nucleus, the control room. It tells this whole giant cell what to do.
3 parts, 3 jobs, 1 cell at 170 millimeters across. That's the ostrich egg yolk.
Remember the holy water balloon? Skin, water, nothing inside. This yolk is the opposite. Skin, jelly, and a control dot.
So, I pick lab bearings. One question first. If a cell can be this big, what's the smallest part still doing a job?
An ostrich egg yolk is one single cell, not many. Every cell has a membrane, cytoplasm, and a nucleus inside. Cell size ranges hugely, from bacteria to a 170 millimeter yolk.